We've got the majority of our judges returned, and at this point, I'd like to ask Dr. Rebecca Richards-Cordum to come up so she can introduce our keynote speaker.
Well, thank you so much. It has been a really wonderful day, and the wonderful things are not over yet. We have one more in store for you. I have really a true honor to present the second of our awards today. We present two Rice 360 Innovation and Leadership Awards. Our first one, the recipient is someone who's very well established in her career and has had a huge impact on the field of global health. But we also recognize individuals who are at the beginning of what we know is going to be a very amazing career. So I have the honor to present our second award to Dr. Nuttada Panpradist. And this award honors all of the contributions that she has made to improving global health equity all around the world. Now, she's at the beginning of her career, as I said, but she is still a very, very effective advocate for health equity. And one of the things I really appreciate about her is the focus that she brings to capacity building and building very meaningful bi-directional collaborations. Now, she's going to tell you a little bit more about her journey, but I'm going to give you a little bit of background. She got her bachelor's degree in petrochemical and polymeric materials engineering in Thailand. But when she graduated and went to find a job, there were very limited prospects for women in engineering, and she couldn't get a job in her field. So she took a role in medical sales and found that she was always thinking up ways to use engineering to think about how she would improve the technologies that she was selling. And so she decided to move to the United States for better career opportunities with the goal of getting a degree in bioengineering and making healthcare accessible and affordable. And she was trying to get herself up to speed. She got a certificate degree in pharmacy technology and a certificate in nanotechnology from North Seattle Community College. She landed an internship in bioengineering at the University of Washington, working with Professor Barry Lutz there. And he recognized immediately what a special person she was and that she really belonged in the PhD program, which she then joined, earning her PhD in bioengineering from UW. And during the time of her PhD and her postdoc, she has not only developed point-of-care diagnostic technologies, but commercialized them as well, looking at new diagnostics for tuberculosis, for HIV drug resistance, and for COVID. She set up new collaborations in Kenya and Thailand. She's just, in fact, wrapping up her postdoc there and preparing to start her own lab as an independent faculty member. She's already won awards for her research and her mentorship. And at Rice 360, we are really thrilled to recognize your accomplishments, especially we are so impressed by your commitment to selflessly creating opportunities for people around you. So congratulations, we're delighted for you to give our closing keynote address. Thank you.
Wow, that got so emotional. Thank you so much, Rebecca, for such a heartfelt introduction. I'm going to try to find it. Perfect. This is the last talk. I will keep it short and simple. So there will be 3 sections. First, it will be a little bit about myself, but now you hear from the first person perspective. Second, it's going to be the journey. What happened today? I hear a lot of amazing technology. What happened after the R&D? I will walk you through a little bit of sneak peek. So hopefully from this journey, you save a lot of your time in the future to not make the same mistake. And lastly, I will show you a little bit about how we as a whole can work together to build local capacity and improve bi-directional collaboration. And just a warning, I actually make some pop quiz. So if you pay attention, you should be able to answer this one with perfect score. My name is Nuttada Panpradist. Sometimes people refer to me as Dr. Panda just to make it more accessible. I'm from Thailand, where I did a degree in petrochemical engineering. Sadly, even though I make it to top 5, male under the age of 35 was what defined on a job description. So I switched my career, taking the job selling medical devices. And oh my gosh, and this is probably the reason why you are here today too. When you started to get the taste of the impact of medical device on global health, you start thinking of ways to make an improvement on that. One of the key challenges I noticed right away is that not everyone can pay for these medical devices. So it's time to learn for me to apply my engineering degree to actually help solve this problem. Sadly, I encountered a second issue, which is inequitable in education. Thailand at that time did not have PhD in bioengineering, and I was not aware of amazing programs like RISE 360. So I made my choice to come here all the way to the US to pursue this education. At first, you might have thought that, oh my gosh, I know everything about global health coming from Thailand, but you wouldn't recognize the differences when you start understanding about healthcare system in the US. We didn't have insurance. We didn't have the system that actually ensured that everyone can access to care. Then I was lucky enough to get engaged in the bioengineering department at University of Washington. And towards the end, I received my PhD and started developing diagnostic for low resource setting. Right away, at the end of the degree, I recognized that it's not just about engineering skill that we need. We need to think about ways for us all to push the technology to make an impact. We need to go beyond the mode that we call pilot and publication, but try to make that impact. And the way to do that is for me to actually engage with entrepreneurship. And you might have heard about this program. I participated in a program called iCore. When I learned about a term called customer discovery, where you learn beyond the need, but you understand the whole key stakeholder of what it takes to take a technology out there. And surprisingly, the same term, it actually exists, but call it differently in global health. We call it implementation science. So I was able to keep writing grants, raise funding to pay for myself to move into the Department of Global Health as the first bioengineer. Trust me, there was a lot of questions. What are you doing here? I at first didn't know how to answer that. I was like, I know that as long as I push through the barrier, I will learn something. And I was right, because I have a team of many people around the world. At the University of Washington, we have, people that come to do MPH from Kenya, from South Africa, from so many countries. And I have a long line in front of my door saying, Nuttada, can we work together to tackle global health problems? So I think whatever you're doing today is amazing by exposing yourself out to learn and experience new things. That was just the beginning. I also recognize that as a postdoc, you don't have access to the lab. So how do you get around with this? So I learned how to partner with 13 different departments at University of Washington to get all of my 10 projects going. And it didn't stop there. I feel like sometimes it's not just a title. I don't want to wait until I become faculty to start teaching. So I accept a lot of honorary positions. So you can see here, I actually teach at 3 universities in Thailand. And the department itself may not actually have some connection, associate medical science, nursing, or even forest biology. But one thing that they have in common is they all rely on a way to detect biomarkers or DNA in low resource setting. So that's how I actually offer my mentorship officially to these students along the way. And to engage myself with the real, world situation, I volunteer myself to be research officer in the Ministry of Health to learn more and become a part of the community. So I'm looking forward to learning more from the Kenyan team here. And just to highlight a little bit about some projects I have ongoing and have done in the past to focus on expanding access to healthcare in the area of diagnostic to address the need for underserved, underrepresented, and underprivileged communities. I'm going to tell you 3 examples of the project I did during my PhD. First, I led the development of the technology called Ola Simple, which is a near point of care HIV drug resistance test that can personalize treatment to match different strains of HIV. We didn't just develop we took it all the way to clinical evaluation. And recently in September, I had an honor to give a podium presentation, and we received the award as the most promising point of care technology for global health by World Health Organization organized meeting committee. Secondly, I was about to graduate after I finished with Ola Simple, but I feel like that was the call for me to take all my knowledge to help fight the battle of COVID-19. So I stick around and co-lead the development of the point of care COVID-19 test that user just only need to collect the sample onto the swab, elute it in the liquid, and then load into this little device, and that connects to the mobile phone. The prototype actually was really well received. The technology then got licensed to the startup that become the commercial product today. Lastly, I could not emphasize enough about ability for us to actually put ourselves out there to connect with people from different backgrounds. I was lucky to participate in one of the seminars in which I was paired with a clinician from Kenya who was very passionate about tuberculosis. She told me one thing that, Nuttada, do you know what? We have gene expert that can detect tuberculosis very well in adults, but in children, they are not able to generate their own sputum. And that's why even though you have really, really good sample, but if the quality of sample is bad, the test fail. And right there, I said, I accept your challenge. We're gonna look into other methods to analyze tuberculosis DNA. Long story short, I identified the gap in the field of the past decade of how people have been detecting TB urine DNA. Proposed a new method that led to a field funding. And to date, we were able to demonstrate one of the most sensitive TB DNA detection from urine sample that has been matched with the 5 requirement. So that's what I did during the PhD. But honestly, what really excited me the most is the new direction that I took to look at the problem of diagnostic, global health diagnostic in the systematic level. I will give you 2 examples. I created a program called University of Washington Thailand Diagnostic Cycle, in which I work directly with the Council of President University of Thailand. It's so special here because sometimes when we come from the small, low to middle income countries, the voice of the president of one university can be quite small. No one may not listen to you. But the power of people that come together to form a council of 36 universities, now you start talking. And this project I would like to share because it could be mapped and transferred to other universities. The way it works is that Thailand gave us a strategic area that they need to improve in global health or other area. They asked us, University of Washington, to identify the potential partnership. And then if you develop the grant internally together, Thailand University will help identify the match organization that willing to pay the money. So now you have 2x of the investment for the get-go. Secondly, this is the way to challenge our global partner to be equitable in terms of contribution to technology development. On top of that, the second example, not all the countries can have that capacity to contribute to money, but it doesn't mean that they don't have dedication and willingness to change. I will tell you more detail about this project later, but long story short, it started with two researchers here in this picture, Ronald and Preston, who came to me and asked me, Nuttada, can we work with TB diagnostic for drug resistance together? And instead of saying yes or no, I said, let's answer this question together. All I'm here is someone who can guide you through the design process. But long story short, the picture you see here is Ronald and Preston, who hold Ola Simple in the first picture that actually detect TB drug resistance. And the second picture with the third picture, one thing I can say is that I don't know what's going on, but Ronald and Preston talking in Swahili. And before you know it, they were able to collect the data that show a successful run of Ola Simple for TB. To me, that is a good way to think about how we can translate not just technology, but the knowledge that could lead to the technology so that we can truly say that we are co-developing the technology through bi-directional collaboration. What I told you in the last slide was just the first domain of my research in the lab. I have other areas that I want to be able to cover today, but it's actually expand toward One Health. So we look at animal health and potential for emergence of infectious diseases. We also try to develop patient-centered method to leverage current existing variable device, but actually engage with patient to really define what is the right design criteria for technology and where the gap is. Lastly, I also work on new chemistry that would allow for detection of sepsis, bacterial sepsis, through using synthetic biology tool. And I have been incredible to be able to partner with 4 countries so far, including Zimbabwe, Nigeria, Thailand, South Africa. Oh, that's 5, Kenya. I apologize with my math. So with that, I think you're ready for the journey of Ola Simple. I will be sharing with you about a little bit about the need and how we actually develop Ola Simple for HIV drug resistance, and what are the type of design criteria, just similarly to a lot of you that present today, but hopefully I can match your shoe because it was amazing this morning and afternoon. And then we'll share you guys the lesson, what that I learned from our attempt to bring Ola Simple to the market. And you see the sign, right? Warning, pop quiz alert. All right. Start with, why do we need HIV drug resistant test? Turns out HIV is one of the most highly mutated organisms. In every cycle that it multiplies itself or replicates itself, it can introduce one mutation. And it's not just that. In patients that actually don't take medication, they're not suppressed, in one day, they can replicate up to 10 billion new organisms. So you can do the math of how many random mutations that can be introduced to HIV genome itself. And to prevent HIV to further mutate, that's why we use the common drug class called NNRTI that stop the reverse transcription process of HIV in order to prevent it to actually further mutate and also suppress the replication of virus. It turns out that only some of these mutations can confer HIV drug resistance if it happened to be at a hotspot. So here is one example in which the base at position 181 changed from A to C. This will change amino acid sequence that make this mutant HIV become resistant to the drug nevirapine. And we are very lucky that thanks to the advancement of genomics science, they were able to identify 6 key mutations that are associated with first-line treatment failure. Therefore, in the US, before even starting everyone with that treatment, they have to do what they call test and treat strategy. They need to know what are the current strain of that person that they have and reflect the proper treatment. Or even after someone starts their treatment already, they have to ensure that the treatment is working. So they do viral load to look at the level of virus. And if someone has treatment failure, they do another round of HIV drug resistant tests. Based on this pretty comprehensive testing and treating, I can imagine it probably costs a lot. So here's the first quick question for you. In the US, without insurance, how much do you think it costs a patient per month to manage a patient who have HIV? Who think is number 1, 100 to 500? Who think is number 2, 750 to 1,500? Who think is number 3? Who think is number 4? All right, the answer is number 3, but I think you realize how expensive this is, right? And that set us to the next stage. And that exactly what one of the reasons why HIV test is actually inaccessible or not equitable for patient care in low to middle income countries. So what happened is that treatment is empirical. Public strategy would just say everyone will have the same regimen. And if you feel like you're not doing well or that viral load is not doing great, which could be that someone not taking the medication or they actually develop new resistance, they just switch the treatment. But something that really sad is that it's not just diagnostic that has limitation. In this setting, in patient one lifetime, you might experience only 2 regimens, first and second. So if you're not using the drug properly, this could cause some problem. So the second question I have for you, what do you think this problem could cause from using this empirical treatment program? Do you think that it can cause poor treatment outcome to each patient? Who do you think number 2, transmutation of HIV/DR, drug resistance? Who thinks it's number 3, undermine effective of the current drug? Who thinks all of the above? And I think we have the class A here because that's exactly what you predicted. It turns out after drug has been rolled out for some time, even before treatment, 1 in 4 adults already, found that are tested to be positive with HIV, already determined to have drug resistant strain. And in kids, it's actually 3 in 4 infants. Therefore, to respond to this, the WHO say, if countries have more than 10% of HIV/DR to this new old regimen and NRTI, you need to switch to the new regimen for integrates or adopt the HIV testing for each patient. So before we move on to the next one, I have a recap. Are you ready? What is the biomarker for HIV/DR? Is it gene insertion? Is it single nucleotide polymorphism? Or is it DNA methylation? The answer is 2. Second question, what enzyme caused HIV genome to evolve rapidly? Who think it's A, reverse transcription? Who think it's B, protease? Who think it's 3, integrase? The answer is A, reverse transcription. All right, last question. What is true? Oh my God, is it going back to GED test, right? Was it true about HIV/DR and HIV/DR test? A, all single base mutation are linked to HIV/DR. B, HIV/DR emerges most rapidly when patients take treatment regimen. And C, DR test is used before treatment initiation in the US. All right, the answer is C. Wow, everyone did really well. So after we define the need, the next thing to define is what could be our unique contribution. Absolutely what you all have done today, identify existing solution, right? And see how you could contribute uniquely. This is what we envision. Instead of sending a sample to read for all sequences in a centralized lab, we want to develop a decentralized test. And this is exactly what picture I used in my business plan competition 8 years ago. Pretty funny that I saw a lot of picture today. It's amazing, much better than my picture. We want to develop the vision of the test that can improve speed, simplicity, and affordability of HIV/DR test to enable use of personalized treatment. To do that, we first have to define what is the technical requirement. The test itself needs to be specifically detect those 6 key SNPs that I told you about, and also tolerate any potential mutation in orange right here that could cause most of the technology progress to fail. It also needs to detect down to 10 to 15% of mitochondrial variants. This is something that's different from COVID, because when we talk about strain of COVID, you just say alpha, beta, right? For HIV, you can have what we call quasi-species, which is a mixture of all the mutation, and you have to detect subset of that to determine the right medication. We rely on a technology called OLA Simple, which actually can detect down to 2 to 5%. OLA Simple can enable high sensitivity using a technology that you might have heard, PCR, that converts just a few copies to billion copies. Then we use highly specific enzyme called ligase that can actually conjugate the allele-specific probe only when the base at the juncture match. If it doesn't match, ligation will not happen. And if you use different type of the label at the end, you will be able to detect only ligated product and report back if someone had mutation of HIV or not. We partner up with Dr. Lisa Finkel, who pioneered OLA in such a way that enable highly sensitive detection of HIV. She actually tried to successfully implement it in different settings around the world. But the issue that we encounter is that the turnaround time is very slow and also require a lot of installation of equipment at front. It can cost up to nearly $10,000. So that's where the idea of OLA Simple begin. And we have currently like 3 versions, but I'm going to show you just our very first minimal viable product of MVP, in which OLA Simple was designed to detect therapy treatment resistant of HIV. You can see here, the only issue that we try to address is number 1, we want assay to be set up easily. So instead of everyone having, who in this room set up PCR before or heard of them before? Yes. So you need to thaw reagent and you add different volume, right? So to bypass that, we actually develop lyophilized or dry reagent that all user need to do is to add a sample and then rehydrate those samples. So just simply like how you make instant noodle, that kind of way, but more, a little bit more precise. And then use lateral flow detection to enable visualization of results. So at the bottom here, you will see that if you see the bottom band, that means a person have mutant. If you see the top band in the middle, that means it have wild type control. And in some occasion, if something go wrong, such as like there's new genome sequence that we didn't accommodate for, you will see ligation failure as a design that we know that, okay, it's time to redesign our test. Instead of publication, we actually did a soft implementation in which I did what you guys did today. I literally took OLA Simple prototype and presented it before the WHO committee at one of the conference. And that was the experience I never forget. Because as an engineer, I feel like the technology will never be ready. I feel like I could make a lot more simple into everything automation. But until I talk to clinicians here that represent clinicians from different countries, I learned that it's okay, Nuttada, to have the test that take 4 hours because oftentimes these patients have to show up and actually get consulting for their adherence before they actually switch their medication. But instead, they actually raise other questions that I or we as engineers didn't think much about. For example, the first question is, would this test work with my population? And does it actually raise the inclusivity of the test? Because sometimes if we design a test that only work with the US, we target certain subtype, right? But HIV is a pandemic, so you have to make sure that the test still be inclusive across population. The second question, from the last picture, you might saw that OLA Simple still have a lot of moving pieces. And is it common to ask, can it be done by first-time user? So instead of doing, we actually take the order and we said, okay, we will develop the interactive software that guide user through the step. And we actually conducted the usability testing. And then last question, and I warn you, this is a question you will get a lot when you start taking your technology out there. Where can I buy this? Because it takes a lot from R&D to take it to the commercial product, which you will hear a little bit later. So instead, we actually engage with the industrial manufacturer and potential users throughout the process. Here is the result of our first feasibility testing. All we did is actually recruited 41 participants at the University of Washington who didn't know anything about OLA Simple, and they were handed to blinded DNA. Some of them don't even know how to use pipette, so I show them 5 minutes and walk away. And they just follow the instruction on a software guide. We were surprised that those combinations enable our first-time user to achieve 97% accuracy. And as I was doing this in the US, at University of Washington, my colleague took another set of experiment all the way to Nairobi, Kenya, and we were able to recruit 12 people that amazingly generate a result that as accurate as the one generated in the US, 97%. So at that point, we feel like, oh my gosh, this is our time. WHO just said in 2017, we need HIV diagnostic test if your country cannot switch to the regimen. We identify our manufacturer capability in 2018, and then think about, should we go startup or should we license it out? We partner up with the manufacturer that actually have the ISO standard that could make a lot of tests. we conducted our clinical validation showing 99.4% diagnostic accuracy. And we even did the field study like you have heard before at 97% accuracy. We finalized the kit into the format that we want and then build a business case. And we at the time identified Mexico as our target country because they are the middle income country and the government said they really need this. And then we actually conducted a study and compare our result with World Health Organization certified lab in Mexico and show that our assay has an accuracy of 97.4. All of this seemed to be all right, right? But spoiler alert, today, Hola Simple is still not commercialized in Mexico. What do you think is the reason behind that? Do you think it's because we are unable to scale the test? Do you think because Mexico have unclear regulatory pathway? Do you think it's because we made it through, but at the end, there's no local uptake? So I won't give you the answer, but I will share with you some of the lessons I learned from this. Pathway to the market, a few lessons I learned. The first challenge is the fast changing landscape of technology could be a challenge to invest. We see example of COVID-19. We were at the point of the peak of the pandemic where everyone said, we want COVID-19 tests. A year or two later, the policy changed. They no longer provide insurance for COVID-19 testing. And now if someone wants to conduct a test, you have to pay out of pocket, right? Unless you have really special insurance. The similar situation happened for HIV. Somehow we tend to forget that HIV evolved very fast in terms of its genome. We believe that a new drug will solve all the problems and completely ignore the practice that accidentally introduced, exposure to HIV drug resistance. So this had already happened before the era of DTG or integrase. The WHO said it's time that we need HIV drug resistance because now the global population have around 10% resistance. They switched to the new regimen and said, for the country that you don't have, please use HIV drug resistance. We think, oh my God, we're ready. And by the time that we actually got through the ready stage for regulatory in Mexico, Mexico successfully negotiated the new drug and said, we don't need your test anymore. We're going to switch to the new drug. And guess what? 2 years passed by. Now we are starting to see the increase of prevalence of resistance to DTG. So this is a problem that ongoing that I believe that it involves education to policymaker to never put an easy way out and just say we can always switch the drug because the drug take a very long time to develop, right? The second one is about scale up production. It's very easy in the lab when we make N=3 or N=5 and write a publication, right? But if you think about, you have to do this 1,000 times, your design process itself need to be parallel, need to be able to be scaled up. So we actually developed live light reagent and lateral flow strip that actually very straightforward. And we were able to make a lot in the lab. But the issue is actually not just making a test. How do you finalize them into a form factor that's ready to use? This is the real bottleneck because we have to recruit the whole lab to put the label and barcode and everything together. So ending up, sometimes we will factor the cost of the test. We cannot just think about the cost of reagent, but have to think about all the manpower that gone into building the test and also QC the test. So this is what happened. We learned that it's a lot of work to actually put the kit together, not just the design process. And if you want to actually, work with the system that built it in the lab, you need to go to quality management system and lock in your design. So we decided that at this stage, it's probably best for us to go with the company that already have ready to go capacity. So these are some considerations when it comes to manufacturing partner. We first actually partner with the company, local company that at the time they were small, but now they exploded after COVID-19, super rich, and probably could not work with us anymore. But we were able to collaborate with them to transfer the protocol that we work in the lab. And in 2 weeks, we were able to get 2,000 strips that transfer from their company to our company. But at the same time, we still need to figure out how to make dry reagent, right? And here is the spoiler alert. If you continue in this path, you actually could find a partnership that the other company that don't just care for the business, but also care for the global good and investment in education. So I was lucky. Actually, that was me participating in this competition and met the CEO of the IEH lab, who were just never say, Nuttada, don't worry, just sign the NDA, we'll make the test kit for you and we talk about money later. So I have been very lucky and I'm happy to make any connection if you are interested. But the lesson I learned is that for me to be that lucky to get a team to work with me, I need to be able to convince the industrial partner to want to invest with the technology. I have to think about what are the new process, what can be done manually, what can be automated. And at the end, figuring out the regulatory pathway that can carry out the study. And there are many more questions that sometimes as an engineer at the beginning, we didn't think about. For example, who needs this test? And who's going to run the test? How many diagnostic tests would be needed for a certain setting? Would it be the same across the globe? Or one setting in Kenya say one thing, but then when I take it to South Africa, it say other thing. That is a real challenge for global health. How can we make our tests universally accessible? And I'm going to end with the last lesson here before I talk about the last piece is when you go out and hear, I need this, it's not enough. You have to ask further, a more important question, who will pay for this? Because that will drive the whole technology innovation to impact. And who will pay for this would depend on the settings. Even in the US, we get funded through Ministry of Medicare and Medicaid. But when you talk about international setting, it will be other organizations as well as the Ministry of Health. With all this, these are 3 key takeaways. If you have a pen or something, take notes. It's very challenging for us to have to work in a market that changes really quickly. But it doesn't mean that you can actually forget your dream, but just put it aside. And what I do is I will continue to raise the funding, focus on those non-dilutive funding. Don't give away your technology yet. We were able to keep writing the grant for Ola Simple, and now we have around 620 million just on the same platform and continue to improve. And then I use the word platform, which means we are just ready and wait for the right technology need. For example, when COVID came, we were ready to actually transfer our knowledge to make technology for commercialization, right? So this is one way to do it. One thing you may notice is that technology is intended for use in low to middle income countries, but all the process of making, developing is all done in the US. I'm going to give you another example of how we can flip the table and make a difference. This is my postdoc work where I start to develop collaboration that I consider bi-directional by directly working with local Kenya researcher and key stakeholder to develop Ola Simple for tuberculosis. It all starts with this question. Someone asked me, visit Ronald and present, Nuttada, how can diagnostic be sustainable so long as we are waiting for our donor to tell us which test to use? And they can stop this funding anytime. And what are we going to do? And the test is so expensive if we have to pay it ourselves. And at the end, how using the technology that developed outside the country could impact our economic and social growth in the country. After COVID, a lot of these voices have grown louder and louder because people really think about how they can actually contribute in tech development and take care and contribute of their own problem so that they can prevent endemic to become pandemic. And the approach I took is that I switched the order of R&D with implementation. I start with need assessment, followed by implementation plan in the country, and followed by R&D that fit the need of that setting. So first, I asked the team, Ronald and Preston, you never done this, right? Can you just help me answer these 3 questions? Where is the need? Where is the gap of current technology? And where does Ola Simple stand? And to my surprise, you know what? It took them a week to come back with very in-depth information because They are the real expert. They know what's going on in the real world, and those are not published data. So they came back with this need that they said, oh God, 10% of children death in Kenya due to tuberculosis. And we need a test to look for rifampicin and isoniazid that actually already, cause resistance in 12% of new cases and 10% in previously treated population. And just to give you a point of reference, this threshold is exactly when HIV committee decide it's time to change the drug. But the problem with tuberculosis and antimicrobial resistance is that there's no other drugs. They have not have so many regimens. So I guess you stick with this first line regimen forever until you have a tool that tell you to switch. And they also identify for me that actually tuberculosis happen to have point mutation or SNP, which allow Ola-Simple to work as well. So that in terms of the need for clinical use. Then they went on and asked them next question, gene expert is everywhere. Where's the gap? So they start to map this complicated treat. I think June will be able to share later how complicated TB treatment that designed by Mr. Pell. It's like, I was confused, but this is a simplified version. The bottom line is that even though a person is tested positive for TB, they may not get that treatment started right away because in some high risk or when they define as having rift resistant, they have to actually send a sample to centralized lab to do a comprehensive first line, it's called first line live probe assay first. So then you have point of care test, but you don't have actionable information. So they say, yes, Nuttada, there's some room for Ola-Simple. If we can actually use this in hospital lab and try to make everything simple with simple thermal cycle that these days cost $500, we might have something that we feel like is our technology. It's something we can use the off-the-shelf reagent to do the work without relying on outside. So I asked them the next question. All right, you convinced me, there's a need there. How do you go around about implementing this in the country? What is your regulatory pathway like? How did the test can get manufactured at scale, not just 1 or 2 or 3 tests, and who will pay for it? So we're still working on that, but we were able to partner with the team at KEMRI, Kenya Medical Institute. Fill in the rest for me, thank you. And they actually identify potential partner where they already start doing lateral flow strip there. And also we think if we are able to raise some funding, we might be able to build in the lifelines technology there. And at that time, all the people that have worked so far are all from Ministry of Health. And today I'm just honored to get to know all the biomedical researcher leader here. So we look forward to talking to you more. And throughout the process, one thing to note is that we engage with national TB program that tell us exactly what it would take us to get through the regulatory process in country, because they usually evaluate these tests at the very end of the product cycle. And then at the point, they said, no, there's a lot of issue. So, but they didn't have a choice because at the end, it depends on who pay for the test, right? But if they are going to take the ownership of it, they said, no. We need a warm-up session to know what's out there. We need to be educated and get ready for the implementation. The last part is R&D cost. When we get a lot of funding in the US, it's very easy to use any approach to develop the test. But how do we go around about building this test or research and development in this setting? So I will break it down into 3 steps. First, this is exactly how we did for Ola-Simple HIV. We do in silico design to develop the probe that target the key mutation predictive of at least 95% of TB resistance. The next stage, I usually just jump into doing the lateral flow test, but guess what? The cost for the probe itself would be around 1,000 for mutation during R&D. So if you were to detect 10, that 10,000 itself, how is that even possible for people in low to middle income country to use that, right? So I have to tweak around and use mail-based analysis that bring down the cost to around, $50 per test, still very high because we actually don't make the probe ourselves. And then the one that actually work well get transferred to the final lateral flow test. So you can see some of the result here that across of the mutation here, you can differentiate between mutant and wild type. Just to share with you, this is a timeline, and it's truly accelerated when we work closely with people locally. I start from ideation when I took Ola Simple to Kenya back in March 2022. We keep meeting over the weekend. We successfully got the funding because these days funders also want to look at something translatable and make impact. And we have a team that have all expertise to tell us exactly where it will be used. So we got the funding, not very big, it's only 100,000. All the team recognize that it's small funding. They refuse to take the salary, but what they want is to be able to be co-inventor. This is what I meant by money is not everything. If you can communicate and collaborate in such a way that people are willing to work together to build something that's meaningful to them, that's the team you want to build around. Then we went through R&D. We already conduct on-site demonstration like you saw in the last picture. And we're now ready to test some clinical validation, followed by the follow-up R01. And throughout every single step, we meet with local researchers and engage with Ministry of Health. So everyone is all on the same page and ready to move at a fast pace. Before I leave, I would like you to take home with these messages. Bi-directional collaboration with local researchers indeed can help you accelerate tech development because they tell you exactly what is the right product for that context. It speeds up implementation because it helps you think what you need to develop to make it scalable and go through successful regulatory process. We can increase user uptake because we already engage with people who are the key opinion leader and help us decide what is needed for that setting. And lastly, we will be able to transfer this technology knowledge. I would like to promote on a few areas. We need to increase equity in medical research and promote decolonization. I would like to say one more time that local researchers in low to middle country are very coachable. What they didn't have is the lack of exposure like what we have today. Imagine if we have ability to coach them, they would be so excellent and ready to take on new challenges in global health. I really care about local capacity building because I believe this is the way for us to truly partner with low to middle income countries to give them autonomy that could be the pathway for true sustainability. And speaking of credit, I mean in the form of authorship of paper, we discussed how we can co-lead or co-last author, talking about potential co-licensing that they feel ownership of the technology. And speaking of credit, I have one person to thank too. I share the opportunity that I will be giving keynote address at RISE at Nigeria at one of the sites that partner up with NEST 360. The clinician told me that, Nuttada, the only way that I will feel like I'm a part of this, you gotta wear a Nigerian outfit and you might see this is exactly what she gave me. So I hold my word to that, even though it's not my style, but I do it. And it's not just that. Throughout my career, I have many people to thank that got me this far. But the group of people that are so motivated that even though I traveled 50% of my time last year, they finished the work. And last year, these are the students that took on 10 projects and we presented at national conference successfully. And I don't have a chance to talk about these projects. So please go on to this website and you can download the abstract there. And lastly, I would like to thank all of these labs that give me access, even though I'm not trained in their lab, but they are my amazing collaborator that I look forward to working with them for more. With that, thank you so much for your time.
Now have time for a few questions.
We'll get the mic working.
Did everyone just like ready to accept the award, right? Nuttada, can we get through this? Yes.
Everyone's blown away. Seriously. What's next for you? If I can ask, in 5 or 2 years, that's an amazing and few years you have a response. Where do you see yourself in 10 years? What are the big challenges you want to take on?
The key limitation is that we don't give enough opportunity to researchers that are so talented in low to middle income countries. So I start with what I can offer. So on the weekend, I would just make phone call with them. They participated for free, try to build a grant together. And one thing that's really hard is that I did everything alone. And I was blown away when I ran into Rise and heard about Rebecca, and I feel like, oh my gosh, you already established a lot of things at Rise 360. So what I look forward to is that we leverage from each other establishment. I don't have to build anything from scratch. We can collaborate, and I would volunteer to even teach here if I had an opportunity. So just I think, just to emphasize, it is important to give equal opportunity for researchers around the world, because that is the human right. Everyone have the right to learn and respond to their curiosity. The country have the right to build their own technology because they know the most. And by diversifying manufacturing process, it would drive not just addressing to healthcare, but also promote their social and also economics. So I think this is the only way. We cannot just keep saying that, oh, we just need a few group of people, donor in the US, to decide what the world needs without listening to what the world really needs. And I think all together today, if we can think about a few ways we can work together, not just me, come to me and come to everyone to work together. I think that would be the second message. Work together to make change. Yep.
Yes, Letho's right. We are blown away. You've answered many of my questions, and I'm commendable that you've managed to break that barrier. Not only you, us as Kenya, we tried to do it alone. During COVID, we were very ambitious and we tried to do it alone, and we discovered it's very difficult. So that's why we are here. We're partnering with people who are ahead so that we're not trying to replicate, but trying to grow together and collaborate. Yeah, so I'm glad you managed to meet Kenya Medical Research Institute. They're very good in terms of when it comes to regulatory pathways, because they've done a lot of that. And yeah, you're in good hands. And next time you're in Kenya, look for me and we can collaborate also in my area. So thank you very much for that presentation. And I don't have any question. I just wanted to say thank you.
Thank you.
Hi, thank you for that presentation. I just had a question about because I remember when you discussed the difficulty with what happened in Mexico and how it was hard to fulfill your project's dreams. So I was just wondering what advice would you have when we try to conduct ourselves with policymakers, because we know sometimes governments don't want to listen to the science. So is it just practice? Is it just being brave and trying your hardest? What advice would you have?
Yeah. I'm glad you asked me this question because I have just the right answer for you. I happened to meet with one of the policymakers in Uganda. She came to us and said, I want to know more about technology development. Don't come to us and say, we need an IRB right now. We need to do the regulatory. We need to be warned what you guys are working, number one. We need to catch up the speed because they are not even equipped on catching up. So a good example would be the beer team, right? That do the testing. The user don't even know what they have capacity for. So by engaging those people early, don't think about them as other team. They are a part of the team because they also share the common goal of pushing global health technology to the finish line to make impact, right? So if we engage with them early and learn from them, ask them exactly what you need, how do we make this faster? In fact, it actually could speed up your regulatory process. So for example, there are most of the technology. Has anyone in this room know WHOP qualification process? Yes. So you will have to do validation across multiple sites and get approval, right? But those are the type of the technology that need to be funded by PEPFAR or other type of technology. By working with different countries, I learned more about different countries. For example, South Africa say, we pay for our own HIV tests by government. So an opportunity to commercialize that would be one of the targets. For example, if you can work directly with the Ministry of Health there and learn what needed to actually push to regulatory process, you don't have to go to WHOP qual, but you can just work with the Ministry of Health there to build the technology, build a local capacity for manufacturing, etc. So don't look at them as an outsider. They are a part of our team. We just need to work together and learn different language. Yep. Mm-hmm.
I have a question for you.
Thank you.
Thank you so much for your speech. I see that a lot in the global health realm that we have academia partnering with government, but I was wondering, what would you think about some of these larger companies, pharmaceutical and biotech? What do you think they could contribute to further develop technology in these low resource settings?
Very good question. I had that question myself because any type of collaboration I think of, I always think about why it's a two-way street, right? How can we create something that could be our partnership? So I would think that for the drug company, they would probably want to make sure that their drug lasts for a while before their new drug actually come out, right? So what might be the way to do that is potentially think about compendium drug model in which, okay, to sell the drug, I also sell diagnostic with you. And the country would make sure, the country that is a part of this whole Colosseum can think about, oh, I only want to work with this drug that come with the free testing. So I think I'll be open to all the discussion to think about, how we can work together to benefit not just the profit of the company, but make the global impact before that. And I'm not sure if you heard about Mistletoe Foundation. I'm actually the fellow there in which they actually focus on how we can think about social and human entrepreneurship. So wait for that. Maybe in 3 months, I have the answer for you better than this. Yeah.
Thank you.
Yeah. Thank you.
Are there any other questions?
I have a question for you. Would you like to collaborate with me on manufacturing? I heard.
That might be an exaggeration, but okay, I would love to talk to you about it. That's sort of what my question was. So I work with a lot of people who aren't in graduate school right now. Maybe they're trying to get into graduate school, or because of financial reasons, they're not in graduate school, or they're trying to get into medical school or something. And a few people around the world who have no opportunity to go to graduate school because of the nation they're in or something like that. What I would really like to see is for us as a community, find a way to allow what we might call the DIY movement, the do-it-yourself movement, or the maker movement, or the less credentialed would-be researchers to participate in some way. Now, obviously, they're not going to be participating in the same way in your lab that your students are in the lab. But I wonder if you could comment on that.
Amazing you asked this question. I thought about that all the time. In fact, I wouldn't be who I am without that opportunity, right? As an international student coming to the US, and just one person see something in me and give me that opportunity. I'm able to stand here and share your story, right? So I can share you a sample. During the COVID, there was one student from India reaching out. I want to get into grad school. How do you go around about this? So I said, oh, we need someone to develop algorithm to detect the lateral flow strip and classify them. She just worked late night, and before you know it, for 3 months, she was one of the person who present at national conference in the US and got into grad school, because at that point, she have learned what it's like to do research in the US, because that's a key gap, because culture is number one. People were trained differently, especially in the country I'm from Thailand. Student is not center. Student is passive. You're supposed to just listen, memorize, and recite. There's no room for curiosity. So if we can flip the table, encourage them to say more, speak more, contribute more, I think that's the first step. And then say that, the sky is limit. There's nothing that can prevent you to contribute. So just to go along that, we can think about, can we start with even pairing the undergrad student at Rice or other university to contribute and teach the technology knowledge that they have? And at the same time, they can contribute the expertise that they have, even through the, not everyone can travel to Malawi through on-site, right? But we can generate something that don't treat ourselves as a tourist. We need to build a partnership that long-lasting, that willing to check in all the time, not just when we feel like talking, right? So that's number one. And number two is that there are a lot of resources that university put out from Rice, from collective university in the US, that oftentimes people don't even register to go through. If there are platforms that are free, we should actually exploit that and consolidate them as a free resource that can lift them up so that they will be at a level that can contribute effectively. Yeah, I think that we all have a lot of ideas. So if there is a way to set that up, I will sign up as #1 participant.
Thank you so much for a really, really wonderful and inspiring talk. We are just delighted that you were able to join us today, and thank you for the work that you're doing.